All right, sounds good. Well, thanks so much for the invitation. This is a delight to do. So, hi everyone, I'm Brian Perry. As you can see from my title slide, I am a professor at Cal State East Bay over in Hayward here in California in the Bay Area. And I thought I'd tell you a little bit about how I ended up coming to Mycena as my first fungal lab and how I ended up becoming a mycologist. And it all kind of started, I was an undergraduate at San Francisco State. I actually went there as an art major, and a semester away from graduating with a degree in photography. I changed my major to biology, took some prerequisite courses where I met this gentleman, Dr. Dennis Desjardin, who's a professor there, he's emeritus now, he's retired. And he came in and gave a guest lecture on fungal biology and diversity to my general intro bio course and basically just blew my mind. I was completely amazed by fungi, these bizarre, fantastic organisms, and I couldn't wait to take his courses. So I took the prereqs, finally ended up in his course, and by the end of the first couple of weeks, I knew that I wanted to be a mycologist. I knew that I wanted to study fungi. And he, being the cruel man that he is, decided that when I came to start a master's with him, he would give me the small job of doing a monograph on the genus Mycena from California. And I accepted the challenge, and I've never looked back. I find Mycena to be an incredibly beautiful mushroom genus, both macroscopically and microscopically, which I hope to convince all of you of tonight. And so I ended up doing a master's with Dennis. I did this monographic treatment of the genus, primarily from Northern California. I only had a couple of years to do it, but still managed to document almost 70 species of Mycena from California for that project. And then while I was there doing that, I decided that a PhD was the next logical step. And so I ended up going all the way across the country to Cambridge to work with Dr. Don Pfister at Harvard. And there I switched gears entirely and started working on ascomycetes. And so for my PhD work, I worked on the family Pyronemataceae with one of the world's specialists on that. So I got really great training working with Don and delving into the ascomycete world, which has been super beneficial. And then while I was there, Dennis and I decided that it would be great to get me back out to California for a postdoc. And so we applied for and eventually got an NSF award to study the evolution and systematics of the mycenoid fungi. So I came back out to San Francisco State for a postdoc for 3 years. And that's where I sort of got off on this track on studying fungal bioluminescence. Right when I started the program there with Dennis as a postdoc, he was working with some folks down in South America, and they were describing these new species of bioluminescent mushrooms that just, excuse me, just happened to be mycenas. And so part of the project that I was doing there for my postdoc, this dovetailed with it really nicely. So for the last like 15, almost 20 years, I've been working with Dennis and also the gentleman behind him, that's Dr. Cassius Stavani. He's a biochemist from Brazil. We've been working on the evolution and diversity of bioluminescent fungi. And so that's been a really big part of my research program for a while. Some of you probably heard me talk about that in previous talks. And then, of course, once my postdoc ended, I had to get a real job, and I got really lucky and ended up replacing one of my other mentors, Dr. Don Hemes at the University of Hawaii at Hilo. And I was out there for about 4 years. Unfortunately, it was impossible to make both my career and my wife's career work. So she and my son came back to California, and I commuted for about 2 years between the Bay Area and Hilo until I finally ended up landing a position at Cal State East Bay in Hayward. And I've been there ever since. And so it's been great. I've had great students. We've got decent facilities. So it's been a good fit. And so while I'm there, continue to work on Mycena and a lot of other projects. And I'll be honest and let you folks know that when I was starting my PhD and deciding to become a mycologist, one of the reasons I did it was because I wanted to find a job that allowed me to spend as much time outdoors as possible and also allowed me to travel. And so I've been incredibly lucky and incredibly fortunate to hook up with the right people. We've been able to secure funding. And as part of that, I've been able to travel around the world to do field work. And so over the last 10, 15 years, a good amount of my field work has actually been in tropical habitats, a lot of work in the South Pacific, some in Micronesia, some in Africa. So a lot of my work on the mycenoid fungi over the last 15 years has actually been from tropical environments. But tonight, what I'm going to talk to you about primarily is mycenoid species that occur in the northern temperate zone. So the best place to start, of course, are, well, what are these mycenoid fungi that I'm here talking about? And I think most of you probably know. That's why you signed up to come see me talk about this. But it's a very large group of fungi that are centered around this genus Mycena. And so we can think about the macromorphological features of the genus Mycena and what are shared with these other groups that we call allied genera or just mycenoid fungi. But what we're saying really is basically they have a mycenoid stature. And what that means is that they can be incredibly tiny, diminutive species to medium-sized species. They tend to have a companionate to almost a conic cap in many cases. They have fragile stipes. They drop a white spore print. Their lamellae are attached in most cases. There's some tropical groups that have a pseudocalarium where they form little structures that almost appear to be free lamellae, but for the most part, they're attached. They have a central stipe in most cases. We'll see in a few minutes that there are some that have a lateral or eccentric stipe. And biologically, they're almost all saprotrophs. There are a handful, or at least one, I should say, plant pathogens, but there's probably more pathogens and parasites hiding out there. So those are the basics of sort of the macromorphological features of the mycenoid fungi. But when we get into it, we see that it's really quite a diverse group. So we can have things that are incredibly small. This is a Mycena up here in the upper middle part. And the scale on that is probably about 1 millimeter in diameter. So incredibly diminutive, tiny species. The one right next to it is Mycena overholtzii, which is one of the largest Mycena species in the world, right? So that's pretty much a big, chonky mushroom that you can go out and collect. But also notice that in amongst these mycenoid fungi, we have things with lamellae, but we also have things with pores. And so this is Phylogeny manipularis or Mycena manipularis, whichever you like to call it. This is what it looks like in the dark when it bioluminesces. So it's one of those species. And this is what it looks like during normal light. And then, of course, we have things like Favolacea, this blue one down here. It's not the greatest photograph, but you can see, hopefully, that there's little pores on the hymenophore of that species. And then, of course, here in the bottom middle, we have Pinellus stypticus. And this is one that is a very tough, almost leathery fruit body that has a lateral stipe. So very un-Mycena-like, but as we'll learn in a little bit, it actually is a Mycena. And then, of course, we have all these other groups that we've moved out of Mycena over the years because they differ in some of the morphological features that we'll be talking about. So let's talk about Mycena itself. So here we have this group of fungi that are centered around the genus Mycena, and these allied or putative allied groups are things that have been at one time or another thought to be either very similar to Mycena, perhaps part of Mycena, or at least closely related to Mycena. So if we delve in and look at the macromorphology of Mycena, it's this basic structure: white spores, delicate small fruiting bodies, mycenoid stature, so having that delicate stipe and a companionate to a conic pileus, attached lamellae, and for the most part, the central stipe. What's really important, of course, is the micromorphology when we delve into this group. And so what you're looking at here, this is a diagram from one of Alexander Smith's book, Mushrooms, Their Natural Habitats. And I'm going to go through and talk about a number of the micromorphological features that are unique to Mycena and the mycenoid fungi. And then I'll come back a little bit later in my talk and I'll show you lots of photographs and photomicrographs of these as well. So this slide always confuses folks. So what are you looking at there? Well, Michael Coe has a nice little sort of diagram here. So imagine that we took a mushroom and we just cut this perfect chunk out of the pileus and slid it out such that we have the context or the cap of the mushroom. On the very top, we have that outermost layer of tissue. We call that the pileopellis when we're talking about the microscopic features. And then, of course, you have the lamellae, and they're sort of dangling down here. So imagine that we zoomed in and just took this middle lamellae, and that is what you're looking at in this slide. So you were looking dead on at a mushroom gill, if you want to think of it that way, that has been sliced in half. So if this were a mushroom gill, it'd be coming out of the screen directly past you and then going into the screen in that blade-like fashion. And so within Mycena, what we see is that microscopically, they typically have a pileopellis, which is that outermost layer of tissue that you can see right on top up here. That is a cutis of radially arranged hyphae. Well, what the heck does that mean? So a cutis is a layer of filamentous cells that basically are starting in the middle of that mushroom cap and running in a radial fashion towards the margins in all directions. And so you get these long filamentous layers of cells just sort of laying on top of one another. And that's what you're seeing here. So going from left to right in this image, you'll see that there's these filamentous hyphal strands here, and they're all sort of stacked on top of each other. And then in the uppermost portion here, these are little side branches that are coming up and branching into sort of antler-like structures. And this is a really common type of structure that you'll see for the pileopellis tissues in a Mycena. Okay, and so we refer to that as a cutis, and this will become important in a little bit. Right underneath that cutis, we have another layer of tissue that is also really important and unique to primarily the genus Mycena and some other genera, but almost all northern temperate Mycena species have what's called a hypodermium. And this hypodermia, as you see in the diagram here, notice that there's these sort of swollen, big round cells here, which are laying over the top of these more interwoven, sort of narrow filamentous cells. That layer of inflated cells is referred to as a hypodermium, okay? Almost all Mycena species that I'm aware of, at least in the northern temperate zone, have a hypodermium right underneath their piliopelis. And that tissue is also dextranoid. And what dextranoid means is that when you put Melzer's reagent on it, which is an iodine-containing stain, it causes those cells to turn red or reddish brown. It's reacting with a polysaccharide in the cell walls, and it causes a red reaction. So kind of hard to see in this slide, but if you look where my cursor is, you can see there's these big inflated cells up in here. That's the piliopelis that you see, sort of the little dirty looking stuff on top of it. And then underneath it here, this would be the actual gill. So that gill is laying flat on its side and we're looking straight down on it, okay? So there's that dextranoid hypodermium, something that is always a great way to determine if you have a Mycena, is looking for that dextranoid hypodermium. And then we get to things like spores. So within Mycena, within everything within family Mycenaceae, we're going to see that the spores are smooth, they're thin-walled, and they're amyloid, meaning that they stain bluish to bluish gray in that Melzer's reagent. So in Melzer's, two different types of reactions, or actually three different types of reactions. You can have dextranoid stains where you get a red to a reddish brown color. You can have amyloid stain where you get a blue to a blue-gray. Sometimes it can even be almost blackish blue if it's really strong. Or you can have no reaction, which would be non-dextranoid and in amyloid. And we'll talk about that a little bit later as well. But within the genus Mycena, in a strict sense at least, and within the family Mycenaceae, what we're going to see is that all species have these smooth, thin-walled amyloid basidiospores. And then finally, we get to the fun part. Okay, so underneath the microscope, I think that Mycena is maybe even more beautiful under the scope than it is macroscopically. And that's due to the presence of these sterile cells that are intermixed with the basidia. So again, to orient ourselves to this diagram here, this layer here that you're looking at, you can see right here, this is a basidium. That's the cell that is the site of sexual reproduction in the fungus. So here we have the spores being produced on the sterigmata. And you've got a bunch of immature basidia and then a few ones in the drawing here that have spores on them. So those are mature. And then intermixed with those basidia, you see these long sort of fusiform cells. And those are sterile cells. They don't produce spores. They're just intermixed with those spore-producing basidia, but they're sterile. And they potentially serve a number of different uses that we're probably not really certain about, but they're really unique in Mycena, and they are consistent from one individual to another within a species. So they're taxonomically really informative within this group. And every single species of Mycena in the Northern Hemisphere, I think, I'm correct on this, has chylocystia. Chylocystia are the cells that are on the lamellar edge. So this is the part of that lamella that's facing the ground. And along that edge down there, you get these clusters of these sterile cells. We call those chylocystia. On the sides or on the face of the lamellae, we get the same types of sterile cells, and we call those pleurocystidia. And so we're distinguishing between the types based on their location. And to show you what some of these look like, this is Mycena hematopus. This is the same species that's actually in that drawing there. This is what it looks like in real life. And so here you can see this is the edge of that lamellae. And so all these sharp little spines that are sticking off are all these cells that are chylocystia. And in this case, they're stained red because I've put them in something called Congo red. It stains their cell walls red so that there's more contrast in the microscope. And then here we see them in Melzer's reagent. So this is that iodine-containing stain. And you can see there's a little bit of a reddish reaction here. So that would be a dextranoid reaction to the tissue right there. But notice that the cystidia, the chylocystia sticking out, are in amyloid and they're non-dextranoid. They're not staining blue and they're not staining red, okay? And then over here, this slide over in the lower left-hand corner is just this slide after I squashed it out. It's what I've done here is I've taken a pair of forceps or an eraser and squished the microscope slide to make the tissue break apart, just so that I could see the individual cells. And I go in and I measure those and draw them and photograph them, obviously. Okay, So those are Cheilocystidia, and it turns out that they are one of the most taxonomically informative micromorphological features in most Mycena species, and indeed in most Mycenoid groups. All right. So in addition to Cheilocystidia and Pleurocystidia, there's two additional types that we might encounter within Mycena and the mycenoid fungi. Those are Pileocystidia, which are sterile cells that arise on the pileus, and they'll be differentiated from the cutis hyphae. And then there's Caulocystidia, and those are sterile cells that occur on the surface of the stipe. So that outermost layer of tissue on the stipe surface is referred to as the cortical hyphae of the stipe. And sometimes those cortical hyphal strands will give rise to these differentiated sterile cells, and we call those Caulocystidia. And this is what they look like. So to orient yourself to this slide, this is a mushroom stipe that is running up and down. So imagine this is a mushroom that's standing at a slight angle like this. And then coming right off of the edge, you see these long filamentous sort of tapered pointed cells. Those are Caulocystidia. Now, if you've ever seen a mushroom with, excuse me, with a magnifying glass or a hand lens, and you've looked at that tissue and you see what look like little tiny hairs coming off the surface of the stipe, what you are actually seeing are Caulocystidia. When you put it under the microscope, you'll get these amazing structures that are really fun to visualize. All right. So that's sort of it for a gist of the main micromorphological features that we'll think about within Mycena. And as I said before, I'll come back towards the end of the talk and I'm going to show you a lot of photographs of these features in a bunch of different species. So be patient. We'll get to more pretty pictures in just a little bit. But what I want to think about now is sort of the diversity of mycenoid fungi and give you a little bit of a synopsis about that and talk about what our current understanding is with regards to the evolutionary relationships amongst Mycena and all these other mycenoid genera. So the best place to start is basically thinking, what is it that we know right now? So if we go and look at the number of species of just the genus Mycena that have been reported from primarily the Northern Hemisphere, what we see, actually globally as well. Singer in 1986, and in his Agaricales, A Modern Taxonomy, he accounts for 309 species in the genus Mycena globally. Okay, so not that many when you think about it. Then Moskis Doranis came in 1992. He did a conspectus of Mycenas from the Northern Hemisphere. I'll say more about that in a few minutes. He accounted for 201, so just over 200 species from the Northern Hemisphere alone. Kirk et al. In the Dictionary of Fungi from 2008 estimates that there's probably 500 species of Mycena globally. But those of us that work on this group think it's probably quite a bit higher than that. Okay, if you go into Index Fungorum and you just look for the number of epithets, the number of species names for Mycena, you get almost 2,500. Now, of course, many of these are going to be synonyms. Many of them are going to be varieties or invalid names. But it suggests that there's a lot out there, right? So there's a lot of Mycenas out there that probably still need a lot of work. And as we all know, there's many, many regions of the world where we've never gone to survey for fungal diversity. Many Mycenas are incredibly small and easy to overlook. So our feeling is that Mycena is probably pretty poorly documented for most regions of the world. And there's probably well over 1,000 species of Mycena out there. Within Mycena, what folks have done traditionally is using morphology, both macro and micromorphology, is they have broken up the genus into infrageneric sections just to try and get a handle on the diversity in the morphology. I'm not going to say a whole lot about the infrageneric sections simply because molecular data doesn't support most of them. I'll mention two later on in my talk that are supported pretty well by molecular data. But for the most part, all these sort of generic things that we've created within the genus don't hold up very well to molecular sequence and phylogenetic analyses. And then, as I mentioned earlier, there's over 60 genera that at one point in time have been considered either part of the genus Mycena or, excuse me, closely related to the genus Mycena. And I simply refer to those as allied mycenoid genera. So these are going to be things like Hemimycena, Strobilurus, Biospora, a lot of these things that are, mycenoid in their stature, but have other pretty distinctive features that separate them from the genus Mycena. All right. Oh, question?
We had a question from maybe a slide or two back about what magnification were you using? Maybe tell us a little about the
Yep, absolutely. And I'm sorry, I have magnification on the other images. I did not put it on this one. This would be at 1,000x. So this would be under oil. for this image. A lot of the Colocystidia on Mycena species, some of them are several hundred microns long. So you can, even at 200x or 400x, oftentimes you can visualize the Colocystidia. Most of the Cheilocystidia that you're going to see in these images, these are also at 400x. And then if you were to see these ones here, these are at 1000x. And so my apologies, I should have put the magnification there. I do have it on the later slides.
We had a follow-up question about the cystidia. You said they're diagnostic. Paul is asking, does it separate to species or just tell us that they are in Mycena?
Yeah, no, definitely we use them for species level differentiation. So we look at the ornamentation, we look at the shape, we look at the size, and that's of course taken in conjunction with a lot of other features, but the cystidia are really the prominent feature in Mycena. You start looking at through Mycena monographic treatments, you'll see that they spend a lot of time illustrating, drawing, or photographing the cystidia. So good question. All right. And so now if we think of the mycenoid genera, the vast majority of them are treated in three separate families. So the Mycenaceae, Syphilaceae, and Porotheliaceae are where we're going to see the majority of mycenoid genera that I'm going to be talking about this evening. There are a few other ones that have, at one point in time been considered closely related to Mycena that are falling out in other areas, but the vast majority of them are in these three families in our current treatments, at least. All right, and so when I think of the classification and diversity of the mycenoid fungi, what I really think is, well, other than Mycena, the mycenoid fungi include really two main groups. So we're going to have species that are not currently treated in the genus Mycena, but probably should be. And then there's also species that used to be treated in the genus Mycena, but do not belong there and have been moved out. So they've been segregated out of the genus Mycena, either into new genera or into existing genera. And so what I want to do is I want to sort of separate the talk into looking at both of those groups. And so what we'll start with is we're going to look at both the Mycena species, so those of Mycena sensu strictu, as well as species that are not currently treated within the genus Mycena, but as you'll see in a few minutes, really should be. Okay, and to do this, of course, we're going to look at phylogenetic trees, everyone's favorite. And so what I've done here just to orient us to these figures, this is a phylogenetic analysis that was just included in a review paper that we published a couple of weeks ago. And this was a review paper on the evolution and distribution and diversity of bioluminescence, of fungal bioluminescence. And one of the analyses that we did in the paper was looking at the Mycenaceae. So looking at family Mycenaceae and looking at the bioluminescence as it is spread throughout that group. And so when you're looking at this tree, all the species names that you see that are in green, those are all species that are known to be bioluminescent. All the ones that are just in black font here, those are all things that either are not bioluminescent or have not yet been reported to be bioluminescent, just because we probably haven't looked. But that's kind of beyond what we're with here. What I want you to focus on in these trees is looking at, so the little gray box over here is showing you what portion of that large giant tree we're looking at. And it'll change as we go from one figure to the next. This is a maximum likelihood analysis for those that are keeping score. And this is based upon two genes. This is ITS and LSU, so ribosomal markers. And this is probably the largest Mycena or Mycenaceae phylogeny, I should say, that's been generated and published to date. Okay, and so it's pretty eye-opening what we're going to see here in a minute. So if we look at this upper group, you'll notice that we got all these Mycenas. Everything that you see in this tree is a Mycena species. You see the blue arrow pointing to Mycena tenerima here, so some familiar ones to some of you that are into collecting little tiny Mycenas. Mycena tenerima up in the upper part there, so this would be section Saccharifery for those that know these groups. And then notice just moving our way down the tree, everything else in this is a Mycena species. However, when we drop down to the next part of our tree, which we show here in the gray box, so this is going to be the group that is sister to that big group I showed just above, notice that we're starting to get other genera. So we've got Roridomyces here. We've got a Rosinomycena that Dennis and I described a couple of years ago, which I guess we got it wrong. It looks like it's actually a Roridomyces. And then moving down, we see Cruentomycena. I should do this. There's Roridomyces for you. So many of you sure know this species. It's the little mycenoid thing with the snot layer all over the base of its stipe. That's Roridomyces. Cruentomycena, which was described probably about 15 years ago by Peterson and Hughes and some other folks. And it's embedded right within these other mycenoid genera. Then we get down to things like Pinellus stypticus. So if we look from here all the way up, we have a whole lineage that is all Pinellus as well as Dictyopanus, which some people just accept as a species of Pinellus. And then below that, we have Mycena, so another mycenoid genus here. And this again is one of those really diminutive species. This is probably about 1 millimeter in diameter across on that fruiting body. So again, we're seeing Mycena species intermixed with Mycena, Pinellus and Dictyopanus, Cruentomycena, Roridomyces, and again, some more Mycena up here. And then if we drop down yet again, so to the next major lineage in our tree, we get to all the poroid taxa. So here we have Favolacea. This is Favolacea thwaitii. Looks very similar to Favolacea calocera or Favolacea cladopis, which is the orange one that we think is an invasive species that we're finding around the world, including here in the Bay Area. It's been found locally. But looking at this, what we see is that we get this really nice, fairly well-supported lineage that's all composed of poroid mycenoid species. So Favolacea here. We drop down a little bit further. We have Phylogeny manipularis, which again is one of our poroid species that is strongly bioluminescent. This is probably the most commonly collected and photographed bioluminescent species in the world. And then again, below that, we get back into more Mycena species. And then finally, we get to the base of the tree. And so just like we saw in the uppermost clade up here, when we get to the lower clade, it's comprised entirely of Mycena species until we get to the outgroup. But what's important about this one, oh, and just to say there's some familiar things. So there's Mycena haematopus up here, if you know that species, is if we look at the very base of the tree, so sort of zooming in right down here at the bottom, we get Mycena gallericulata. And Mycena gallericulata is the type species of the genus Mycena. And this complicates things a lot. Well, it actually doesn't. It makes things really simple, but it's going to be tough for some folks. So these are analyses that are based on 2 markers. We actually have larger analyses from our smaller sampling that are based on multiple genes. And we resolve a similar situation where you are getting the type of the genus of Mycena coming out in a basal position in the evolutionary tree. And all these other genera like Roridomyces and Mycena and Favolacea and Cruentomycena and Dictyopanus and Pinellus, they are all embedded in the phylogeny, intermixed with other Mycena species. So as we've seen with other groups recently in the last decade, so Cortinarius recently, someone went in, folks go in and they will break up an entire genus into multiple genera based upon molecular data and usually some morphological data as well. With Mycena, it's going to be very difficult to do that because we have standard mycenoid things like Mycena gallericulata, which just looks like a big gray Mycena with normal gills at the base of the tree. And then if we make our way up that tree, we see that all the way at the very top, we have more standard Mycenas there. So the genus Mycena spans the entire phylogeny, and then embedded within Mycena, we have all these other genera. And this tells us mainly something that we've known for a long time, and that is that Mycena, as it's currently circumscribed, is a non-monophyletic genus. We've known for many years that Mycena is polyphyletic. We get all these other things that are falling out in with Mycena when we do these phylogenetic analyses. So no huge surprise there, okay? What our data are suggesting is that rather than recognizing or continuing to recognize all these genera like Cruentomycena and Dictyopanus and Favolacea, we should actually be moving many of these into the genus Mycena. And in many cases, they'd be moving back into the genus Mycena because many of them are simply things that were a Mycena at one point in time, but based on morphology, it was separated out and put into a different genus. Okay, so our data is suggesting that there's a simple solution to this, and that is simply to move all of these genera, all 7 of them, based on our sampling, back into the genus Mycena. And I know there's going to be questions about that.
The first question is from Jerry Bloom. He is asking about the name of the reference paper.
It's evolution. I should know this. It's a paper. I'll send it to you, Jerry. You can get it on my website. It's there. It's an open PDF. Anyone can download it, but it's the Evolution, Diversity, and Distribution of Bioluminescent Fungi. It's published in Journal of Fungi just about 2 weeks ago.
What's
All
your
right.
website again?
Website is just perrymycolab.com. You can also go to Journal of Fungi, and it's in the current issue. It's actually the cover story of the current issue. There's a nice photograph of Phylogeny manipularis on the cover. And so at any rate, what our data is suggesting here is that Mycena is a very large, currently non-monophyletic genus that harbors a huge amount of morphological diversity. So what we intend to do at some point is likely merge all of these genera into Mycena, and then we can either recognize the well-supported clades like Favolacea and Rhodomyces either as subgenera within Mycena or as additional sections within Mycena. And so we'll hopefully be doing that within the next year or so. But this begs the question. I went through all those morphological features with you about Mycena, all the micromorphology that unites that genus. And hopefully you're asking the question, well, do we see that micromorphology occurring in all of these other genera as well? And the answer is yes and no. Okay, so I'm going to run through some of these examples in just a few minutes, but if we look at a group like Rhodomyces, we're going to see that it has a very different type of pileopellis compared to a normal Mycena species. Same thing with Rhizinomycena. If we look at Pinellus, we see that it has different tissues. It's got a very different type of structure than a normal Mycena species. All these poroid things. So lots of variation there. The only feature that really unites everything within Mycenaceae right now is that they all have smooth, thin-walled amyloid spores. And to some degree, most of them have some type of dextrinoid tissue in the pileus, whether or not it's the pileus context or the hypodermium or the lamellar trema. So amyloid spores seems to be the only really uniting feature there, and the rest of the morphology is highly variable within this group. All right. So hopefully that gets you all intrigued and you want to go out and start collecting Mycenas and Mycenoid species and bringing them back and trying to key them out and identify them. And to do that, you're going to need some pretty specific resources. And I have my list of favorite books. So there's lots and lots of papers out there that are really important, but there's also a series of really monumental works that should be in the library of any mycenologist. Okay. And the first one is going to be Alexander Smith's monograph on North American species of Mycena, published in 1947. Now, in this, Smith includes a lot of things that we no longer consider to be species of Mycena, some things that actually have like roughened spores that are way outside of Mycena as we know now. But it's a phenomenal amount of work that Smith did, especially if you're working in the Pacific Northwest. Smith spent a lot of time up in Northern California and Oregon and Washington in the 30s, the late 30s and early 40s. And there's a huge amount of great descriptions of species and descriptions of novel species from that area as well. Then I mentioned earlier, Ariamaschi Staranis in 1992 published his compendium. It's a 2-volume set, and it is called Mycenas of the Northern Hemisphere. And what it is a collection of papers that he published over the years and finally bound it together into a single book. Came out in 92, and I would not have been able to do my master's thesis if it was not for this book, because what Maschi Staranis did was he did a type study of every single species of Mycena that was described from the Northern Hemisphere. So he actually went in, borrowed the material, did full microscopy on it, and wrote full descriptions. Absolutely monumental piece of work. Not sure how big the screen is you're working on, but if you're lucky, you might even be able to go to Half Price Books and get a copy of this for 20 bucks, like I did. So actually, my graduate student found 2 of these in mint condition sitting in Half Price Books. So I got both of them for 40 bucks. They've been out of print for quite a while, so they can be challenging to find. But I know that there are scans of this book out there. So ask your mycological friends. I'm sure you can get yourself a copy. Uh-oh, cat got in here. Hold on one sec. You get out of here. Sorry about that. All right, and then some guy by the name of Perry.
Oh, sorry to interrupt there. Somebody had a question. I thought this would be a good time to hop it in there.
Perfect.
I think it was from a little bit further back. It was from Doug asking, why did some species of Mycena lose luciferase activity?
Ooh, why did they? Well, go read the paper, Doug. No. So what we know is that it doesn't take much to lose luminescence in these species, and I'm happy to talk more about this at the end as well. But what we see is that we know that bioluminescence probably arose a single time in the evolution of the Agaricales, and then it's been lost multiple times. So all it requires is really a single simple mutation and one or more of the genes that are part of the bioluminescence pathway, and you can render the whole thing non-functional. And so for many of these, there's probably a ton of Mycena species out there that do bioluminesce, but we've just never observed them doing so. Either they do it at really low levels that human eyes can't detect. We need to use a camera or a luminometer to do so, or they've simply lost the ability. And so that's something that we're trying to do right now, is document as many species as we can to determine how many truly are bioluminescent. So, good question. All right, and so I wrote a monograph for my master's degree in 2002. So that is really focusing on Northern California, and that is available for free. So if you go to mycoweb.com, which is Mike Wood's website, great site if you haven't been there before, go check it out. But Mike has copies of a lot of monographs that you can download there, including my master's thesis. So you can get a copy of that if you want. And then Giovanni Robic in 2003, and then in 2016 published 2 volumes on the Mycena of Europe, volumes 1 and 2, that are absolutely stunning. Beautiful photographs, beautiful line drawings. It's in Italian, but he also has descriptions and keys in English. So certainly worth picking up. They're still available. You can still get those. So definitely great books. And then most recently, Arne Arntzen and Tom Laso published the genus Mycena sensulatu, and it's really focusing on Northern Europe. And in this case, they're looking at Mycena, but they're also including things like Phleomana and Atheniella and other genera that have been segregated out from Mycena. So it's sort of Mycena in a broad sense. So again, really great book, wonderful photographs, nice line drawings. So certainly worth picking up. You can still get that one as well. Then, of course, there's websites. So as I mentioned before, MycoWeb is awesome. It's really, that one really focuses on California fungi, but Mike has tons of great information there, great photographs, descriptions of many species. Wonderful site to go check out. Arne Arntzen also has a Mycenas of Northern Europe, which is sort of the online companion to the Mycena sensulatu book. Very nice, great photos, keys, wonderful things like that. The Pacific Northwest Key Council has great keys to Mycena for the Pacific Northwest on their website, so definitely worth checking out. And then I also have keys on my website. And so if you go to perrymycolab.com, you can go click on the Mycena of California link up at the top, and it will open up a key for you. That is a key to Mycena species from California, but keep in mind, many of these are going to also occur up into Oregon and Washington and British Columbia as well. And what I've done here is, whoa, it's getting way ahead of myself, sorry, is I've worked with Mike Wood. So as you're working through the key, if it's a species that there are photographs and descriptions of on Mike Wood's Myco website, there's simply a link there. So as you're keying something out and you get to a name and you wonder what that thing looks like, you just click the link, it'll open up a separate window. And Mike and I are currently working on that. So ideally we'll have many more mycenoid species added to MycoWeb very soon. So check that out if you want. And then finally, iNaturalist and Mushroom Observer are both excellent resources, especially iNaturalist right now with all the fungal diversity survey work that's going on. There's a huge amount of information being put into iNat, and there's sequence data in many cases. And there's really insightful discussions about the taxonomy and systematics of these collections that they are documenting. So definitely check out iNaturalist if you haven't yet, for the mycenoid fungi especially. All right. And then the most important resource probably if you want to start identifying mycenoid fungi is one of these. So at some point you are going to hit a wall unless you have a microscope. So when you start using keys, whether it's my key or Moskowitz key or Arn's and Tom Laso's key, you're eventually going to be asked things like, what are the size and the shape of the Chylocystidia? Are the spores amyloid? What's the size of the spores? Are there clamps on the hyphae? So you're going to need a microscope to really delve into identifying Mycena species, right? So it's going to be something that, you know, it might be hard to overcome if you've never used a microscope. I encourage you to go try at some opportunity to play with one. Things like mycenoid fungi are incredibly beautiful under the scope, and I think you would probably really enjoy a chance to check it out. So hopefully you have options and opportunities to play with microscopes at your local mycological society events. If you're interested in buying a scope, there's lots of opportunities out there for that as well. There's wonderful scopes made by Modic that are, you know, semi-affordable. What I usually recommend folks do is try and find a used scope. but find someone who knows microscopes and can help you discern, decide whether or not the price is right and whether or not it's a good scope to buy. I'm happy to serve as a resource for anyone that needs that. So microscope's going to be important. And then finally, it used to really be the domain of research institutes and academic institutions to sequence the DNA of fungal specimens for research and doing things like systematics and taxonomy and phylogenetic analyses. But as Bob said, the times there are changing. And for the last decade or so, what we have seen is that there is a huge increase in the amount of folks that are not affiliated with universities or research institutes that are becoming incredibly proficient and talented in both sequencing individual genes as well as entire genomes of fungi and other organisms. And a lot of this has to do with the fact that this technology is much cheaper now, and it's also just much more widely available. So places like Countercultural Labs, which is here in the Bay Area, has done an amazing job of providing opportunities to train folks on doing fungal systematics and sequencing of DNA. You already know about iNaturalist and Mushroom Observer. They're both excellent places where people are posting a lot of the sequence data that they are generating. And of course, we have wonderful databases like NCBI's Nucleotide Blast, where you can take your sequences and blast them against the database to see if there's close matches. And there's multiple databases out there besides just NCBI. So we're in a period of time right now where, lots of folks can do things at multiple levels mycologically. So it's never been a better time to be a community scientist in the mycological circles. In addition to that, we have wonderful things like the Fungal Diversity Survey that are sponsoring projects to document fungal diversity all around North America. And then here in California, more locally, we have institutes like CIB, the California Institute for Biodiversity, that is providing funds to local mycological societies to do fungal diversity studies. And that's to both generate specimens, but also to generate DNA barcode sequences for these as well. So lots and lots of great opportunities out there nowadays. So in my mind, we have multiple tools that we can use when it comes to trying to identify and study diversity of mycenoid fungi. Question?
Yeah. Lisa G is asking if you have any tips on sourcing Meltzers for those without current sources. And in the same thread, there is a little conversation going on about chloral hydrate as well. So if you want to comment.
Yes. So the best way to get Meltzers is to befriend someone at a university who has a license to buy chloral hydrate. So I'm constantly giving Meltzers away, but my university doesn't have a license for chloral hydrate. So we're pretty limited. Oftentimes I will, basically trade with mycologists at other universities that have it. I've heard rumors that you can buy chloral hydrate on eBay. I won't say any more about that, but I do know that lots of folks are making Meltzer's reagent at home. So there are ways out there to acquire it. If you're really hard up for some, reach out to me or other mycologists, so we can usually provide you with some. Okay. But I know that's a tough one. All right. So thinking about that. So I think now we've established, this is a huge amount of diversity. You now know what the tools are that are required to really delve into identifying Mycena and the mycenoid fungi. So now let's go and look at some of these things, right? And so what I want to start out with is looking at species that are not currently treated in the genus Mycena, but likely should be, as well as looking at some Mycena species. And when I was going through and deciding which species to include in the talk tonight, I decided that I would try and give you as many, number one, that I had good photographs of, or what I think are good photographs, ones that I have micrographs of, so I can show you what the cystidia and other tissues might look like. But really, they're also ones that I think you can easily learn to recognize in the field, even without the aid of a microscope. So these are ones that have pretty distinctive macromorphological features, which you could really easily confirm your identification if you did have access to a microscope. And so we'll walk through some of these. And so we'll start out with Mycena haematopus. I think this is one that most people probably know. This is the bleeding bonnet, although I hate that term bonnets, but this is Mycena haematopus to me. It is one that has a red latex, as you can see in the photograph here in the upper right. This species has specialized lactiferous hyphae. They're specialized hyphal strands throughout the entire mushroom that contain this latex that is this very dark blood red color. So when you break the mushroom or damage it, or an insect comes and takes a bite out of it, it exudes this latex. And Typically, you'll find a lot of it at the base of the stipe, but oftentimes, depending on how fresh the specimen is, you will actually see it up in the pileus as well. So it'll exude latex when you break it. Many of the populations will have marginate gill edges, meaning that you see white gills there, but the edge will be pink to red, but not always. It's a pretty variable character. They typically grow in these fasciculate clusters. So this would be a cespitose clusters where the bases are all sort of attached to one another and they're all arising from the same point. And then the telltale one that I always teach to my students is that if you look at Mycena hematopus, you'll see that the pileus edge extends beyond the lamellae. So you get this little lip of the pileus extending beyond, and it typically becomes sort of frilly or eroded. And that's a really distinctive feature of Mycena hematopus. So if you've got something which is this beautiful sort of rose color, you've got reddish latex that it exudes, and that little margin that extends, chances are you've got Mycena hematopus. If you look at the stipe surface as well, you'll notice that they have all these little hairs all over the surface. So when you look at this under the microscope, you are going to see beautiful caulocystidia all over the surface of the stipe there. And this right here, this is just showing you the chylоcystidia. So the first photographs I showed you all of chylоcystidia were actually from Mycena hematopus, but it forms these really cool sort of fusiform to fusoid pointed cystidia. And these are going to be chylоcystidia and pleurocystidia in this case. So that's Mycena hematopus, really common in the northern temperate zone. So I'm sure most folks have probably seen this one. And then one that's really near and dear to my heart is Mycena californiensis. This is one that I played a role in resurrecting this name when I was a first-year graduate student working with Dennis. There's a species in California that used to go by the name Mycena elegantula that was known, it was actually described from Pasadena, and it is a latex-bearing Mycena that has this sort of reddish orange latex. However, there was a collection of a species that was described by Berkeley and Curtis as Mycena, well, as Agaricus californiensis, and then Sicardo made it Mycena californiensis from the 1830s, and it was collected at Mare Island Naval Yard as part of the Wright expedition, and it was sent back to England, and it was described as Mycena or Agaricus californiensis. And then the type was lost. And it was for many, many years, it was assumed that this was a nomen dubium, meaning that it was a dubious name with no specimen to back it up. And then Dennis Desjardin discovered that the Farlow Herbarium at Harvard actually had a paratype. And so we were able to get our hands on that, looked at it, and determined that indeed this and Mycena elegantula were the same exact species. And so Mycena elegantula is now a synonym of Mycena californiensis. And as Elsa Valinga has confirmed for me, this was the first mushroom species ever described from California. So pretty great that it has the name californiensis. But this is a super common one for us in the oak woodlands, at least here in the Bay Area and throughout California down south. Is there a question?
Yes.
All right.
Sorry to interrupt.
That's okay.
There might be a couple. One is asking about, does anything other than Mycena sanguinolenta bleed? I mean, within the Mycena group.
Yes, so this one does. So Mycena californiensis contains latex. Mycena sanguinolenta contains latex. Mycena hematopus does. So those are like the three common red latex containing species. And then here in the West, actually throughout the northern temperate zone, we have Mycena gallopus, which has white latex. And then there's a few others that that occur in Europe and Eastern North America that I don't see them that often, but there's a number of latex containing Mycenas here. Yep. All right. Sure. So this is Mycena californiensis, and it does appear to be restricted to growing with our coastal oaks, primarily Quercus agrifolia. It grows on the leaves and on the acorns and twigs and things like that. But we do see it occurring on other oak species here in California as well. So another beautiful one, it's got these really nice red to reddish brown marginate gills. So nice mushroom to find. And then this is another one which is somewhat similar. This is one that does not contain latex, but it does have marginate gills. And this is Mycena bulliformis. So we described this one, not too long ago, probably 15 years ago or so. This is one that I discovered during my master's thesis work. And it looks very much like Mycena purpureofusca, if you know that species, which I'll show you a photo of that next. But this one is much more reddish brown in color. And it has these sort of gill edges that are violaceous to violaceous brown. If you get it under the microscope, it has these beautiful chyloscystidia. They're these really long sort of fusoid cells. But what's distinctive about this species is that the pileopellis, which is what you're looking at down here, it gives rise to these really big inflated irregular cells. And the term for that is bulliform. So inflated irregular cells is bulliform. So we named this one Mycena bulliformis. Another common one for us around here, I've collected this throughout California. I've collected it up into Oregon and Washington as well. This is certainly one that we have throughout the Pacific and maybe even further east as well. I'm not sure. And then this is a photo of Mycena purpureofusca. This is basically the sister species to Mycena bulliformis, but rather than being reddish brown, Mycena purpureofusca is much more violaceous purple in its coloration and typically grows on different substrates. This one is typically on the bark of conifers, whereas Mycena bulliformis can be on hardwoods as well as conifers. But this is a nice photo. I just took this the other day. So what you're looking at here, looking straight down on the lamellae, and just look at those beautiful purple lamellar edges. Really, really pretty. And then look at the stipe surface here. You see all those hairs? Those are all cholocystidia that would be nice to look at with a microscope. All right, and then a couple of other ones that are really easy to recognize. So Mycena overholtzeri is the largest Mycena in California. It's also one that we find with the spring snowmelt. So when you go up to the Sierra Nevada or the Cascade Ranges, find conifer logs that are partially buried in snow that are super saturated with melting snow. And you will typically find these large clusters of Mycena overholtzeri. First time I collected this, I did not believe it was a Mycena because it can be huge. It looks like a giant colibia or something like that, but it forms these big cespitose clusters. The base of the stipe has this really intense, really dense white tomentum all over the base. Beautiful companionate caps with these nice marginate or striate margins on the cap and decurrent gills, which almost becomes sort of a smoky gray color. So really distinctive, doesn't really look like most Mycenas, really easy to recognize. So definitely that's one that you don't need a microscope to figure out at all. All right, and just a few more because I have some nice photo micrographs of them. This is Mycena rosella. This is one that's really common throughout North America. It's going to be this pink to pale reddish pileus. It's going to fade as it dries out. It starts out much redder, fades to pink as it matures. And it develops these tones also on the stipe and on the lamellae. One of the distinctive things about it is that the sides of the lamellae will develop these little pinkish spots on them. And that's actually due to pigmentation in the cystidia. What's really cool about this mushroom is that the chyloscystidia sort of look like balloon animals or these really oddly shaped things, these weird little protuberances sticking off of them. But the pleurocystidia are a totally different shape. And this is a really rare condition in Mycena. Typically, if you've got pleurocystidia and chyloscystidia, they look the same. There's only a handful of species out there, at least that I'm aware of, that have divergently shaped chylo and pleuro, and this is one of them. So pretty common with us here. I think it gets even more common as you move up north in the conifer zone. So should be pretty easy for folks to find. Mycena maculata, another large Mycena that you do not need a microscope to identify. This one kind of looks a lot like Mycena overholtzeri because it occurs in these big clusters. It has this dense white tomentum at the base, but this one grows in different habitats. It's not a spring snowmelt species. It's a fall species in the mountains and on the coast. And it doesn't have decurrent gills. So if you flip it over, it's not going to have the gills running down the stipe at all. This one also develops sorted stains in age. So both the cap, the stipe, and especially the lamellae will develop these weird sort of reddish brown bruises all over them. And that's what maculate means. So the name Mycena maculata. Again, beautiful mushroom. It's a larger Mycena, probably the second largest Mycena in California, behind Mycena overholtzeri, maybe the third largest. Grows in big clusters on wood. Easy one to recognize. So keep your eyes out for that one.
We have a question or two or three or four. One is like, wait, did you say sorted?
Sorted. I did.
S-O-R-D-I-D?
Yes. So the reddish brown stains that this one develops in age is referred to as sorted in a lot of the older literature.
Okay. Interesting. Thanks, Sarah, for kind of catching that. let's see. Donna asked, do you smell or taste? Do you use smell or taste to help ID Mycena?
I do, depending on how large it is, right? I smell everything, every mushroom I collect, I smell it. And if mycenas are large enough, I will taste them as well. There's very few mycenas that have distinctive tastes, but we'll get into a few in just a minute here that have really distinctive aromas that are it's really indicative of the species. So I always definitely smell them. Yep, good question.
Okay. Now here's a little longer question. It says, thank you for a wonderful and well-organized talk. It seems from work by Eric Hobie and others that mycenas are demonstrating some complex patterns of ecological behaviors or interactions.
Yep.
Although only a few species have been studied, I wonder if you have a sense as to whether these appear to be any pattern related to phylogenetic relationships and whether mycenas may be restricted to wood or found in plant roots.
Yep, good
Do
question.
you have a finally? Do you have a sense of why some mycenas have such large genomes? Okay, that was the whole question.
Yeah, that's a whole nother question. Yeah, so the whole ecology of these is a so we have for years we've assumed that these things are saprotrophic because we find them growing on decaying wood and on leaf litter. In reality, that's exactly what it sounds like. It's an assumption. So we definitely know that in the tropics we get a number of Mycena species forming mycorrhizal relationships with things like orchids. So we do know that some of them are mycorrhizal. They form a specific type of mycorrhizae. Some of them are also pathogens. I mentioned that there's at least one. So Mycena citricolor is a pathogen of coffee. It's a really common pathogen and pest throughout the coffee growing regions of the world. And now we're starting to see that mycenas are popping up more and more in mycorrhizal studies. So chances are they're probably doing a lot more out there than we've traditionally thought. And part of it could be due to this expansion of their genomes. That's pretty amazing work showing that some of these things just have these massively expanded genomes that undoubtedly has to do, we would think, with the ecological roles that they may be involved with. Yeah, so great question. There hasn't been a lot. So Eric Hobby's done a lot of the stable isotope work. And I don't know if he's ever looked at Mycena specifically. I haven't followed what he's done lately, but I know I'm guessing that at least many of them are saprotrophs, but they should be doing more than that out there, would be my guess. Great questions. Yeah.
Okay, the next one is, do you think the Mycena overholtzii of Western NA and Eastern North America are really the same species? The gross morphology seems different.
Yeah, and it gets even weirder than that. So we also get a thing in the fall growing on the coast in the Pacific Northwest that we've called Mycena levigata, which matches at least ITS sequences are identical to the type of overholtzii. So it's a weird one. And I don't know if we have sort of, if this is speciation in action, where we're seeing that these are isolated populations, both spatially and temporally, that are diverging from one another. It could be something like that. But yeah, the verdict's still out. I think there is someone who's going to address that question. I think someone from the Pacific Northwest group was hoping to look into some of these questions about the overholtzii that occurs throughout, North America to see. Yep, good questions, guys. It's great.
There's another one here. It's Mycena strobilinoides and Mycena californiensis sympatric in California, and is the eastern version of Mycena strobilinoides distinct? From Cape Cod's from Cape Cod pitch pine forest.
Yeah, another great question. Yeah, so I was just looking at the distribution of strobilinoides and I was putting this talk together. I've got a picture of it a few slides ahead here. I think of strobilinoides as being, at least in the Pacific Northwest, as being much more of a northern species. I don't typically see it like as far south as where I live. I've got to go a little bit north to find it, like above Mendocino. I might just be missing it though. Mycena californiensis is going to basically it's going to be in different habitats. I think of strobilinoides in the West as being much more of a conifer zone species, like pure conifer stand, whereas Mycena californiensis is always going to be in the Quercus agrifolia or Quercus dominated habitats. But yeah, great question. And then the one in the east, I have no idea. I've never seen strobilinoides from the east, but I'm sure there's probably sequences of the eastern and the western one available. So it'd be pretty interesting to compare them and see how they're looking. But yeah, it's always amazing to me when you see this thing that kind of goes all the way up and over across Canada into like Massachusetts and stuff. And then now it's also known, I think, from Switzerland as well. It's been reported. So cool.
Okay. The next one is back to Meltzer's. In place of Meltzer's, is iodine acceptable in a pinch? There are some conversations in here about where they're finding it in Europe. And a related question came through: is Schultz reagent a substitute for Meltzer's?
So there's other variations out there. There's Lugol's. So I mean, Meltzer's reagent is unique because the chlorohydrate is a clearing agent. And so chlorohydrate is removing any pigmentation from the cell so that the iodine has a chance to stain it more easily. There are versions of it out there that are used, but it sounds like they actually operate slightly differently. So I know there's at least a handful of publications out there where folks have compared Lugol's solution and Meltzer's and then other iodine-based stains to see, if we can get away from using chlorohydrate. And if I'm recalling correctly, I think that depending on the preparation of the tissue prior to the stain, we actually get slightly different results. So you may not get an amyloid reaction in the absence of something like chlorohydrate for certain taxa. So yeah, it's a tough one. But Meltzer's a tiny bottle of Meltzer's will last most people for years and years and years and years. So it's I'm sure you can find some if you're looking for it. In the old days, you could go to a compounding pharmacy and get it apparently, but not anymore. All right. And so someone asked about aroma and odor, and I've got two slides here for things that have really distinctive odors. And there's a whole section in Mycena, section Fragilopedes, which is characterized by having these kind of nondescript grayish brown mushrooms that many of which have this bleachy or this alkaline odor to them. And super common. So I put two in here that are going to be at least the most common ones that I see, at least in the Pacific area. So the first one is Mycena leptocephala. This one's pretty easy to recognize because when it's really young, the pileus is incredibly dark brown to almost black. You'll find them growing they can come out of soil, they can come out of wood or woody debris, but they're typically growing on the ground, not coming out of a decaying log or anything. Very strong alkaline odor. Some people describe it as being like a raphanoid or nitrous. To me, it smells really alkaline. For me, the distinctive feature of leptocephala is when I get it back to the microscope, is I look at the stipe colocystidia. And the stipe colocystidia have these beak-like projections on them. To me, that's the real distinctive feature of this species. The cystidia that it forms are very similar to like Mycena alkaliniformis and other species that look very similar macromorphologically. But leptocephala, these big sort of beak-shaped projections on the colocystidia seem to be pretty unique to that. So that's what I'm always looking for. But again, a really common Mycena with us in the Pacific Northwest. And then a very similar appearing species, even more common, is Mycena capillaripes. And this, going up to Mendocino in the fall, this is probably the most common mushroom. We find it growing in troops of thousands of individuals underneath pines, underneath other conifers, and even redwoods. Really, really common, has this alkaline odor and dead easy to identify because it grows on the ground in big clusters, big numbers, I should say. It has an alkaline odor, and when you look at it with your hand lens, the gill edges are pink. So super easy to recognize this one. No microscope needed. Was there a question?
Here's a pair of questions for you. Yeah, this one I think is a question. Mycena leptocephala present in sphagnum? Question mark.
I couldn't hear the question.
Mycena leptocephala present in sphagnum? Question mark.
Certainly possible. Wouldn't surprise me. I find it. We find it in oak woodlands. We find it in conifer zones. So it wouldn't surprise me to find it in sphagnum habitats. Yeah, but okay. Definitely check the colocystidia.
Okay, that was from Brandon. Glenn asks about Mycena californiense. He said, I thought I heard was limited to Quercus agrifolia, but then I thought I heard that in California it tended to be in conifers. Both, or did I misunderstand?
Yeah, no, definitely. I've never seen it with conifers here. It was described from Quercus agrifolia debris. It's really common in the Quercus woodlands certain years. I have seen it reported from other like coastal and valley oaks here in California, but I don't know about up in Oregon. I haven't seen much about the distribution. I should probably check like INAT to see if folks are bringing it in. But to my knowledge, I've never seen it in conifer habitats, always in oak woodlands.
Lisa G asked if there are any active DNA projects collecting specimens specific to Mycena.
Um, there's me. So I'm always trying to sequence everything I collect. Um, I know people are working on the group. There's a lot of folks that are interested in Mycena, and there's certainly sequencing material that they're bringing in. The Fundus project, so the California Fundus project, every single Mycena and Mycenoid thing they bring in is getting at least an ITS barcode sequence. And then we'll go back and pull out the things that are of interest and sequence other markers for them. So certainly, yeah. And I know Steve Russell, Mycota Labs, he's got a whole project right now documenting fungal diversity in California, sort of picking up where the fungal diversity survey left off. And so he's offering free sequencing of specimens. So definitely worth checking that out if you want to get some material sequenced. All right, a few more that you could easily recognize in the field here. This is Mycena philippes. To me, this is one that I can easily teach my students to recognize, and for me, it's really distinctive because, again, like we saw with Mycena hematopus, notice how the pileus margin extends beyond the lamellae and gets eroded, so you get this little frilly fringe. But it's also incredibly leggy. You get this really, really long stipe and a very small pileus on the top. And so to me, it's a very distinctive looking mushroom. It's pretty pale. Some people think it has a distinctive odor to it, like sort of iodine. If you've ever smelled like iodine, you clean a wound with, it kind of smells like that. Grows on wood. If you get it under a microscope, what you'll see is that the chylocystidea are these beautiful, big inflated clavate cells, and they have these finger-like projections just sticking up in all directions. It kind of reminds me of like an inflated glove or something. So really, really cool under the scope. Pretty easy to recognize in the field, easy to bring back to the lab and confirm with the microscope. And then a few more. Mycena navicola, this is one that we described from the Sierra Nevada. This is one of our spring snowmelt fungi. For years, we call this thing Mycena griseoviridis, but griseoviridis is an eastern species that grows in the fall with hardwoods. This is a spring snowmelt species that grows on conifer debris. And so we finally got good material, got the type of griseoviridis and compared it with our Mycena from the Sierra Nevada and Cascades and confirmed that it is indeed a different species. Beautiful thing, pretty easy to recognize by its coloration. So you get this olivaceous brown cap, you get a stipe that is lemon yellow, and then typically will develop these really dark reddish brown stains towards the base, has really distinctive chylocystidea. And one of the fun things about it is that the pileopellis is completely embedded in gelatinous matter. And so this is a section of the pileopellis. And what you're looking at here, this sort of net-like structure, that's actually all the pileopellis hyphae standing up in this big gelatinous matrix. And you can take a pair of forceps when this thing is fresh, and you can literally peel off the pileopellis off the mushroom as this gelatinous sort of like rubbery layer. Pretty amazing. Not many Mycenas do that. Super common in the Sierra Nevada up into the Cascades in the spring with the snowmelt. So another one super easy to recognize. And its close relative is Mycena epipterygia. These are all within the same section within the genus Mycena, and these ones are pretty morphologically similar to one another. So Mycena epipterygia, again, is going to have this pale yellow stipe, typically has a pileus that ranges from white to yellowish to brown. It's a species complex here. There's been a number of varieties that have been described from California, or sorry, reported from California and the northern temperate zone, and they all seem to intergrade. It's all based on pileus color and substrate. And it'd be great if someone would come through and do some nice, molecular phylogenetics on this group and see how many species we have. But again, really easy to recognize in the field. And then finally, the last two ones we'll look at here. So Mycena pura, and again, this is another species complex. Most folks probably know this one. They're big, beautiful, violaceous to purple Mycenas. Some of them even run the gamut to almost red. Many of them are brown. There's even some pale ones out there. It's certainly a complex. We have no idea how many species of Mycena pura we have in California. Again, it'd be a great project for someone to take on. But these are pretty big Mycenas. They're kind of colibioid in their stature. They kind of look like a colibia or a gymnopus many times. Many of them that we have are purplish or lilac colored. Some people think they have a raphanoid odor to them. We find them in every habitat, and they might look absolutely identical macroscopically and microscopically. That's what their cystidia look like. But when you start sequencing their ITS and other genes, they're totally separate. So here we have a situation where there's probably a whole lot of cryptic diversity hiding under the name Mycena pura. And this is one of the sections that I will mention. So this is section Calodontes. Section Calodontes is one of the only, or one of 2 or 3 sections within Mycena that is always well supported in molecular analyses. And it's pretty easy to recognize it as a cohesive group morphologically, both micro and macro. Yep, question mark.
Question about how to spell raphanoid. What I think smells like a radish.
Yeah, that's it.
Okay. And then the other one was from Anonymous. Any recommendations, tips, or devices to get nice cross sections for under the scope?
Using a dissecting scope, so a stereo microscope to make your sections is really helpful. I work almost exclusively on dry material just because I find it once it's dried, it's much easier. Like handling a tiny fresh Mycena, they just sort of disintegrate in your fingers. So I tend to dry things first on the dehydrator, and then I can section them with a double-edged razor blade much more easily. Practice is what it comes down to. Unless you have access to something like a microtome, it's just come take a microscopy workshop and we can teach you sort of different approaches to sampling. Like if you want to look at the pileapellis versus the chylo or pleuro, we can teach you different sections to make. But at the end of the day, it just really comes down to just practicing.
The question is, do we know if all the Mycena pura complex have muscarine?
That's a good question. I don't. I'm trying to remember what Christopher Bugharder did. I can't remember if they were looking at muscarine content or not. Good question. I don't know the answer to that one. Yep, it's certainly been reported for the species. All righty. So moving on. So the last one I'll have here within Mycena, this is Mycena tenerima. Showed you a photo of this one earlier. And this is a member of section Saccharifery. And again, this is another section that typically can be recovered using molecular phylogenetic analyses. It usually comes out as a pretty well-supported group. And these tend to be the really tiny, diminutive Mycena species. What's unique about them is that they have a universal veil. And when they expand, the remnants of that universal veil are left behind on the pileus as what looks like little sugar granules, hence the name Saccharifery. And if you look at those under the microscope, oftentimes they're going to be these cells we call acanthocysts. And so these big inflated globose cells that have all these little spinuli, little nubbins all over the surface. And these things actually will disarticulate and get washed off the pileus by rain and wind, or a mycologist, touching it. And each one of these little cells has the ability to germinate. So these actually act as asexual reproductive propagules for the fungus. So it's kind of getting a little bit of fragmentation type reproduction out of its sexual reproduction stage here as well, which is pretty cool. Easy one to recognize, small, white, has obviously tons of collocystidia, so little hairs all over the surface of the stipe. Top of the pileus is covered in these carocytes, or these acanthocysts, little granules, and really beautiful, distinctive chylocystidia as well. So that's Mycena tenerima. There's a whole bunch of species in section Saccharifery that they need more work. So Dr. Desjardin did a wonderful world monograph on the group, but we haven't taken the time to really figure out what we have, locally in California or in other places of North America. So another group that could use some work. And then a few others just because they're pretty. This is Mycena corticalis. This is, again, a diminutive species, very, very small. That's probably 2 or 3 millimeters in diameter for the pileus there. Has these great chylocystidia that look like, basically, it looks like a little clavate cell with all these crazy deformed fingers sticking out at the end of it. has globose to subglobose spores, which make it a little bit easier to recognize. Most mycenas have ellipsoid spores. And this was one when I was doing my master's degree, we just didn't encounter it that frequently, and it had only been reported from California 1 or 2 times. And now it's a total weed. I see it every place I go. I see it on oaks, I see it on calisandrus, I see it on sequoias, I see it on Doug fir. Really, really common if you just go out and look for it. So that's Mycena corticalis.
We have a question here from Jack Johnson and Lauren Ray.
Mm-hmm.
What terms do you use to describe the basal discs?
So, I mean, a basal disc is going to be a specific, it's going to obviously be a disc-like structure, and you have to be able to distinguish that from just radiating hyphae. So for me, it's going to be, the basal disc is going to be obviously a pretty thick layer of tissue, whereas the radiating hyphae is just going to be sort of spreading out onto the substrate. To me, they seem pretty obvious. Some basal discs are almost like a little donut at the base of the stipe, whereas others kind of like flatten out and you can actually see the imprint of the lamellae on them. So there's quite a bit of variation. To me, if it's a really noticeable structure that is, persists, I call it a basal disc. If it's just radiating hyphae, I just call it radiating hyphae. Yep.
Thank you. Donna asks, are stipe sections cross sections or longitudinal?
It depends on, I always do a longitudinal, I always do a scalp section of the stipe on a Mycena, just because if you cut it in half, you're just going to be looking at this round thing. And what you really want to do is to be able to follow the hyphae up and down the stipe surface to look for clamps and look for ornamentation and colocystidia. So I always just take the stipe and sort of lay it, flat like this. And I take a double-edged razor blade and just sort of slide it along really carefully and just cut a little piece of the scalp off the top.
Great. Thank you. Amy Honan asked, do you think corticalis range / abundance is increasing?
It's a good question, Amy. It certainly could be, but I almost just feel like people just weren't looking for it before. It's one of those, it's pretty easy to overlook. And I just think there's a lot more folks out there nowadays collecting, smaller mushrooms. Like there's just more interest, but certainly could be expanding its range. We should look at maybe like in Mycoportal, see if there's like more historical collections, outside of California. See. All right, and then finally, here's your Mycena.
We have one more. Can you talk about the difference between Mycena corticola and Mycena melagina pseudocorticola?
Yeah, so you've got to get in and look at this astidia. It really comes down to looking at the spores. So the spores, depending on if they're globose versus ellipsoid or subglobose, looking at the size of the spores. And then for me, getting in and looking at the chylosestidia, I think that's the way I always kind of differentiate between melagina and corticola and pseudocorticola. Yeah.
Okay, I came back because Elsie has a question. Do you think that these mycenas live in the bark of the wood? And where are the hyphae when they are not fruiting?
Yeah, so this is a great question, and they do. So we know that these hyphae persist in the wood. So these things they're not pathogens or anything. Keep in mind that the bark is dead, right? So these are just breaking down the dead wood. You can actually go out and collect bark during the dry season and bring it back and put it in a moist chamber. And species of Mycena will fruit on it. That's actually the title slide. The image on my title slide is Mycena ulmi. And that was one that was reared from American elm bark collected in the Fort Worth Botanic Gardens that we described as a new species during the pandemic. The people that found it were trying to grow slime molds. They were looking for corticulous slime molds, and they had all these moist chambers with the bark in it, and all of a sudden all these mushrooms started growing out of it. And so we assume that the hyphae does have the ability to persist in that bark, and it's probably pretty dormant during the hot, dry months, and then just becomes activated once the cooler temps and the moisture comes. All right. So there's Strobilunoides. We talked a little bit about this one before. This is it's unique within Mycena in the sense that it's a true Mycena species, but it's also very, very brightly colored. And what we'll see in a few minutes is that most of the really like pink, orange, bright yellow Mycenas like this have been moved out into other genera. This one is a good Mycena. It's this really intense red, orange to orange yellow color. It's got red-orange marginate gills, and we find this one, again, the range extends all the way up through the Klamath and Cascade Ranges up into British Columbia, and then it hangs a right and goes kind of across Canada, I think, and even comes down into like Massachusetts and places like that. And now it's being reported from Switzerland as well. So big distribution. Whether or not it's all the same species across, I'm not sure, but kind of hard to miss, and another easy one to identify. All right, and then finally now into a couple of these genera that should be treated within Mycena, right? So here's Rordomyces. This again, we saw this one earlier. Rordomyces rortus is the species that we have here in the West. At least it might be more than one hiding under that name. But small, pale mycenoid fungi, they have this really distinctive gelatinous slime layer all over the surface of the stipe. And this was always considered Mycena rorida for a number of years. And then Rexer in the 90s came along and segregated it into a new genus based on the fact that the Pileopellis is not a cutis, but rather it's a layer of these big globose hymeniform cells. So you get this palisade layer of inflated cells rather than, remember, a cutis is this layer of filamentous hyphae all sort of running in a radial fashion. In Mycena rorida, you just get these big globose cells forming that Pileopellis. So based on that morphological difference, Rexer moved Mycena rorida and 6 additional Mycena species into Roridomyces. And then over the years, an additional 13 species have either been transferred or published as new species of Roridomyces. However, our analyses all suggest that this is just variation. So this truly does appear to be a Mycena. It's just a Mycena that has a different type of Pileopellis. And similar situation for Rhizinomycena. So here we see that this was erected by Scott Redhead and Rolf Singer back in the early 80s to accommodate these mycenoid things that had amyloid spores, but they also have these resinous, often capitate cystidia that look like that. So under the microscope, these things are absolutely beautiful. And you can see this is a great photo here that I got online. You can just see all the cystidia covering the surface of the stipe, the lamellar edge, the entire Pileopellis. So this is probably about 1 millimeter in diameter. These are very diminutive species. You get them under the scope, and you get these often capitate cystidia that are covering every surface of the mushroom. And oftentimes they'll have these really oily or resinous appearing contents inside. So really, really cool. Always a treat to find a Rhizinomycena and look at it under the microscope. Currently 13 species and 3 varieties are treated in the genus, but I'm guessing there's a whole lot more of them out there if we just looked a little bit harder. All right. So now we can finally jump into species that used to be treated in the genus Mycena, but do not belong there and have been moved elsewhere. And so these are going to be some of these are the ones that you probably know many of these already, but these are beautiful species that can be really easy to recognize in the field without the need for a microscope. And so the first genus we'll talk about is Atheniella. We segregated this out of Mycena back in 2012. And we did this based both on morphology and on molecular analyses. So what we've known for a long time is that this species, Mycena adonis, Mycena aurantiadisca, and a few others that I have listed here, when you look at them under the microscope, their spores are inamyloid, so they don't stain blue at all. And they have no dextranoid tissues whatsoever in their pileus, their stipe, or anywhere. And many of them also sort of lack that typical big inflated hypodermium as well. So based on morphology, and then when we started sequencing their DNA and doing phylogenetic analyses, we're able to show that these things fall way outside of Mycena. They're actually over in Cyphalaceae, and we moved them out. So we did that. And since 2012, an additional 7 species have been transferred to the genus or described as new to science. And so the 2 most common ones that we see here in the West are Atheniella adonis, which is this beautiful one here. So it's got this orangey red to pinkish red, sort of coral red color. And oftentimes really pretty yellowish tones to the stipe. We find it in the conifer zone. And if you look at it in the scope, in amyloid spores and non-dextranoid tissues. So pretty darn easy to recognize. And this one I think is even easier. This is Atheniella aurantiadisca. And this is one that when you first see it, the pileus is this sort of like reddish orange color right in the middle, but as it ages, it becomes totally two-tone. So you get this bright yellow disc, and then the margin becomes white. And so this really distinctive two-tone color to the cap. You get this pale yellow coloration to the stipe. In amyloid spores, non-dextrinoid tissues. So again, pretty darn easy to recognize in the field. And then one of my personal favorites, this is Phleomana. So Scott Redhead segregated this out of Mycena in 2013. And again, he did this based on both morphology and phylogenetic analyses. And so this one moved it out because, again, in amyloid spores, non-dextrinoid tissues. So it looks a lot like many, many other Mycenas, but it is lacking in the main features that we use to unite that group. Gene analyses throw it outside of Mycena. And then since 2013, an additional 7 species and a couple of varieties have been transferred from Mycena into Phleomana. The one that we have here in the West, and I think probably the most common one throughout the northern temperate zone, is Phleomana spirea. And again, these can be very, very small, delicate species, typically growing on wood. If you look at their lamellae, they're arcuate, meaning that they sort of come up and then they run down the stipe, almost like a little decurrent tooth, if you will. For me, one of the really defining features is when you look at them under the microscope, the basidia for this species are almost always two-spored. So rather than the usual 4-spored basidia, you get 2-spored basidia in amyloid spores, of course, and non-dextrinoid tissues. And then here at least here in the West, we get this variety called variety Camptophila, which develops these beautiful yellow stipes. So it looks just like the other Phleomana spirea that we see out there, except the stipes are really yellow. However, I just went today to see if anyone had moved this yet, and it still hasn't been moved into Phleomana. So if someone wants, they can go online right now and transfer variety Camptophila to Phleomana and get your name on a species. But kind of silly why that hasn't been done yet. Not sure. But again, pretty distinctive, easy to recognize in the field once you get a feeling for it. All right, and then another one that still needs a home, but it's easy to recognize. This is Mycena oregonensis, described from Oregon, obviously, by Alexander Smith. And can't miss this one. They're very diminutive. They're small, very delicate. The pileus is probably only a couple millimeters in diameter. Bright, bright orange. They grow on both woody debris and herbaceous debris. Get them under the microscope, and in amyloid spores, non-dextrinoid tissues. This one falls out close in our phylogenetic analyses to Phleomana, but it bounces around quite a bit. So this one certainly, I think it's probably will be moved into its own genus at some point, but no one has done that yet. So for the moment, people are calling it Mycena oregonensis. If you look on Mycobank or Index Fungorum, they list it as Hemimycena, which is kind of crazy. All right, folks. So I just want to say thanks for all the great questions and listening. I wish I could say all the photographs were mine, but I borrowed them from lots of folks, including those listed here. So I just want to acknowledge their contributions and say thank you.
That was great. Thanks so much.
You bet. My pleasure.
I mean, I love the not just learning about this genus or the Mycena and its many closely related mushrooms. Your pictures were fabulous, and I think people were really interested in the microscopic elements of your presentation, especially based on the questions. Now, based on that, Donna had one related there asking, is oil immersion, do you use an oil immersion objective lens? Is planar needed or aspherical acceptable?
Yeah, I do use oil. For Mycenas, especially looking at like the pileopellis, for measuring spores, for measuring cystidia and stuff like that, I always go up to immersion oil. All of our objectives are planar, but anything is fine. As long as you can see okay, that's all that really matters. And so, yep.
You're getting a lot of thanks flowing through the chat. If you want to
Thanks
look at that
all.
and see everybody thanking you there. Rodney Webb asks, what is your current count of Mycena species in California? Greater than 70 question?
Yeah, definitely. So for my master's thesis, which keep in mind was like 20 years ago now, I think I had 67 or 68 species that I documented just from, basically from like Monterey north. Like we didn't do much field work in the south that because I was, 2 years I was there, it was really dry. And there wasn't that much material that had been collected by Alexander Smith from the southern part of the state. So I think of my monograph as being like Mycena of Northern California. And now, I mean, I'm guessing that there's well over 100 species in California, would be my guess. And the fungal diversity survey folks are turning up a lot of things that appear undescribed. I didn't get into it in this talk, but we're still applying a lot of European names and concepts to species that occur here that are probably not the same thing as what occurs there. So we probably have a lot of undescribed diversity here in California and throughout North America, really.
Okay, this is, if anybody else has a last question, you got to pop it in now, though. Brandon Roddy is asking how long Metzger's is good for. He's had his stash for 4 years.
No, it's fine. I've had it's as long as you can still put droplets on and it spreads out, it's totally fine. What we always find is that the rubber stopper, rots on the dropper bottle. So we're constantly like changing the dropper portion out. But no, I've had bottles of Melzers that are over a decade old, if not longer, and they're totally fine. I think when I was a student at San Francisco State, I was using Harry Tears. I was using his Melzers, which had been there for decades. So, yep.
Okay. Well, great. Thank you. Was this your last slide?
That's it. Yep.
All right. Well, you did have a lot of thank yous flowing in. You have a huge thanks from us over at NAMA. We really enjoyed this, and we're going to post it online on YouTube in the next couple days.
Awesome.
Good night, everyone.
It was a blast. Thanks all.